All right, so first let's go over a bit of information on symbols for alleles. So previously we used this uppercase, lowercase. So this was the dominant allele right here. The capital letter just depicts the dominant, and then the lowercase represents the recessive allele. So geneticists use different symbols for allele names, and a lot of it depends on the organism they study. So there are some specific allele name schemes for Drosophila, for example, and then there's specific schemes for other situations as well. So kind of the reason behind this is different. So in Drosophila, for example, they had this idea of a wild type because they did a lot of mutant screens. They had this idea of a wild type allele, the natural allele, and then they would name their alleles that they isolated in mutant screens where they actually mutagenized the animals. So kind of the important thing is to get used to these different ways that geneticists name the alleles, and this could either be because of the different organisms, and then also this could be because of different relationships. So not all alleles follow this dominant recessiveness. So an example of this is in Drosophila. So the way Drosophila represents the wild type allele is using the plus symbol. And the plus symbol is pretty much, you can assume, is dominant to the mutant allele. So an example is body color. So they isolated some mutants that affected the body color of the Drosophila fruit flies. And the mutant allele is indicated by this E right here. And then the normal wild type color, so the ebony mutant is controlled by this allele, and then the wild type color, the normal color of Drosophila flies, if you isolated them from the wild, would be gray. And that's indicated by either an E plus or sometimes just a plus. And so, you know, if you wanted to write the three possible genotypes, you could do it this way: plus plus, plus E, and EE. And so this is another way, another example of allele naming where they're using the plus, and the plus is dominant to the E, which you can see based upon the wild type color and then the mutant color phenotypes. So, you know, unfortunately, there's not this like unified scheme where every single system, every single organism follows the same naming convention, and so part of being in genetics is getting used to those different ways. We also have different naming schemes, typically if they don't follow dominance recessiveness, and so an example of this is in these flowers right here. These are called, what are they called? 5 o'clock flowers, 4 o'clock flowers. Some sort of time o'clock flowers. And so, you know, as geneticists, you notice that there are two different colors these flowers are found in. There's this red color and then there's this white color. And these are, again, pure breeding populations, so we know that they're homozygous for whatever gene causes the color of the flowers to be different. And so we're going to name these alleles a little bit differently, and we'll see why in a second. So we have named CR for the red, and then CW for the white. So there's, again, two alleles of this gene, and the homozygous genotype is red, and the homozygous white is white. And so this individual, so if we did a cross between these flowers, this individual can only produce one gamete, same with this one, and so we end up creating this heterozygote individual. And so this heterozygote individual, so this is one of the first surprises, was not red or white. So in the simple dominance recessiveness that Mendel studied, it would always be either red or white, depending on what was dominant to which. But in this case, it's actually a heterozygote is actually a different color, a different phenotype, and it's this pink color instead. And then if we go on and do an intercross, same way as Mendel did, where we took the heterozygotes and self-fertilized them to create an F2 generation. What they found was a 1 to 2 to 1 ratio in the F2s. So red was 1/4 of the progeny was red, 2/4 of the progeny was pink, and then 1/4 of the progeny was white. And so if we just do a simple Punnett square, this makes sense. So this individual now can produce 2 different alleles. So the gametes, there's 2 different gametes, depending on which one of the alleles is packaged up into the gamete. And so we do a Punnett square where both sides of it are a 2x2 Punnett square. And so then if we simply combine it, this is the only way to get a red, this is the only way to get a white, and then the 2 heterozygotes will be pink. So this is an example of incomplete dominance. So if I asked you what kind of relationship exists between these alleles, you should be able to answer incomplete dominance. And so incomplete dominance is the situation where the heterozygote is a mix of the 2 pure genotypes. And so you can see how if you mixed white and red together, you could get pink. And you can distinguish this from a situation of codominance. So codominance is a situation where both parental phenotypes are present. So you can see the flowers have red and white colors within it. So the codominance is when both parentals are present in the heterozygote, and then the incomplete dominance is when the phenotype is a mixture. So again, these are the hets on the bottom, and then these are the pure on the top. And these both would be caused actually, sorry, so this would be the situation where the heterozygote was dominant. So if it was dominant, then the red was colored, and then if recessive, it would be right. But in all of these cases, you assume that the parents are either red, a pure red crossed to a pure white. So these are 4 different examples of relationships that you can have between 2 alleles, and this is the extent of what we'll cover in the class for this. So you can also have situations where you have multiple alleles of the same gene. And so this was a situation when scientists studied the fur color of certain rabbit populations. And so these are 4 different phenotypes. So they noticed that there were 4 different phenotypes that these rabbits could have for fur color. So there was what was called wild type. So these rabbits had brown fur. There was the chinchilla phenotype, where they had black tipped white fur, so it kind of gave the appearance of gray. There is Himalayan, where it's primary white fur, but the animals had black paws, nose, ears, and tail, so they're kind of like highlights of black fur. And then there is the albino, which is the white fur. And so what the scientists discovered is that there was actually 4 different alleles of this gene. So there is a gene called the C gene, and this has 4 different alleles which are named as so. So there's big C, there is CCH, so C chinchilla, little ch, so little c Himalayan, and then just little c. And they discovered that the pure, the homozygous of these alleles would lead to the following 4 different phenotypes. Now, when they do crosses, if they do crosses between these different phenotypes, these 4 different colors, they then also figured out the types of allelic relationships between these 4 alleles. And what they found was that there is a progressive set of dominance. So big C was dominant to 1, 2, 3, all 3 of these alleles. CCH, the chinchilla, was dominant to Himalayan and little c, and then the CH was just dominant to the little c. So one thing that's important to remember when you're discussing these things, so these are 4 possible relationships between alleles, you have to describe this for all of the different combinations of alleles. So for this means that there are actually 4 different genotypes that can have brown fur. So they can either be pure homozygotes or any sort of heterozygote where you have a single C+. So as long as you have a single C+ allele, that C+ allele is dominant to all 3 of the other alleles, and so you end up with brown fur. For chinchilla, there's 3 different genotypes that can lead to this. So there's the homozygous, and then it is dominate to the Himalayan and the albino allele. The Himalayan, there's two different genotypes, so either the pure or if it's heterozygote with the albino, and then the only way that you can have albino is if you're homozygous for that allele. So again, multiple alleles of the same gene. So this is an example where there's 4 alleles of the same gene, and they interact in a dominant recessive fashion. So that is misspelled. So this is not an instance of codominance or incomplete dominance. So you might want to say that, oh, this is a mixture, Himalayan is a mixture between chinchilla and albino. This is not the case. This is not an example of incomplete dominance, because incomplete dominance refers to comparing. You always have to compare the heterozygote to the 2 homozygotes. And incomplete dominance only occurs when the heterozygote is a mixture between the two homozygotes. So in this case, the Himalayan, so this is a case of dominance recessiveness, because if we compare the heterozygote, it has the same phenotype as one of the homozygotes. So hopefully that makes sense. So let's do a quick example of a monohybrid cross. So what happens if we cross a pure breeding chinchilla to a pure breeding Himalayan rabbit? So we cross these 2 rabbits together. So this can only produce 1 gamete, so it can only produce the chinchilla allele, and all of the gametes from this individual are Himalayans. So the F1 would be a heterozygote. And so if we go back to here, we notice that this individual right here is a chinchilla rabbit. So all of the F1s are chinchilla. Now let's say we cross the F1 together and cross 2 siblings together, let's say, 2 heterozygotes. What do we get? So in this case, the F2 is going to be a 1 to 2 to 1 ratio of genotypes, and it's going to be 3 chinchillas to 1 Himalayan. So how do we see that? Again, we do a Punnett square. The F1 can produce 2 gametes. And so we have a 2 by 2 Punnett square, and then 1/4 of them will be this, and then so on and so forth. And so if you completed that Punnett square, you would end up with 1 to 2 to 1, and then you would group these 2 together because they would have the same phenotypes. All 3 of these would be chinchilla. So this looks like, kind of your typical ratio, 3 to 1, but now let's do something a little bit more complicated. So instead of assuming that they're pure breeding rabbits, so we're again crossing the chinchilla to the Himalayan animals, but now we're going to assume they have the following genotype. So this individual is a heterozygote between the chinchilla and the Himalayan rabbit is a genotype is Himalayan with albino. And so now, if we look at the F1 when we cross it, we actually get a non-purebred. Because these are non-purebred, we get a mixture of phenotypes in the F1 generation. And we actually get a mixture of not only a mixture of phenotypes, but also a mixture of genotypes. So again, we can do the Punnett square. So this individual right here produces these 2 gametes. This individual right here produces these 2 gametes. And so there's actually 4 unique genotypes that are produced. And this one right here is chinchilla phenotype. This one right here is chinchilla phenotype. This one right here is Himalayan. And this one right here there is a typo right here. That's supposed to be Himalayan. This one right here is also Himalayan as well. So you end up with this 1-to-1 ratio of chinchilla and Himalayan. Another classic example of multiple alleles in a population is your blood type. So I think most of you are probably familiar with the possible blood types that you can have. So human ABO blood groups. So there's 4 different possible blood types that you can have. You can have an A, a B, you can be AB, or you can O. And so when scientists studied the segregation of human blood types, they discovered that there is, again, a single gene, but the gene had 3 different alleles, and that led to the multiple types of phenotypes. So we have 4 different phenotypes, and because of that, we produce different antigens on our red blood cells. So if you have an A blood cell, if you have an A blood type, then that means you produce this antigen, the A antigen. If you have the B blood type, that means your blood cells has the B antigen. If you have the AB blood type, then you produce both. And if you have the O, then you don't produce any antigens on your red blood cells. So your blood type is an interaction with your immune system. So your immune system produces these specific antibodies that would either recognize the A antigen or the B antigen. And so what happens when your body produces the A antigen is that you actually clear out those antibodies from your bloodstream. So you no longer produce the antibodies that recognize the A antigen. So that would be something that would be very bad because your immune system would attack those red blood cells. But the individuals that have an A phenotype, an A blood type, they still would produce the antibodies that recognize the B antigen. So when you think about blood type, you got to think about both what your blood cells contain, and then also you have to think about your immune system and what they recognize. So this kind of shows what your antibodies are for the 4 different blood types as well. So this is why the O is the universal donor, because you can give O to all 4 of these different blood groups, and because there are no antigens on the blood cells, you never have a problem with your immune system reacting with it. But think about what happens when you take your AB group, and if you transferred blood from the AB group to someone that had the B blood type, they would have these antibodies that would actually attack those red blood cells, and that's kind of what causes the problem. So AB can receive all 4 blood types because it doesn't have any antibodies to worry about, but the O is the universal donor where it can give to all of the different blood types. So what exactly are these antigens? So these are carbohydrate groups that are bound to lipid groups on red blood cells. So these things right here are carbohydrates that are attached on the lipid groups, kind of on the cell membrane. And this is called H substance. So you have this H substance where you have either 1 or 2 terminal sugars are added. So you know, if you're an O type, you kind of have this H substance. If you're the A blood type, you have 1 sugar. If you're the B, you have the second sugar, and then if you're AB, you kind of have both sugars. Is kind of the concepts of how this all works. So the H substance is where the sugars attach to. So O blood types still have the H substance protruding from the red blood cells. They just don't have the sugar groups that are attached to the end. All right, so what are the genetics? So we've talked a lot about the phenotype about your blood type, but what are the actual genetics that control what you express? So what are the genetics that control which of these antigens that you express? And it turns out that this is all controlled again by a single gene, Isoagglutinogen, and this controls what antigens are expressed. And there's actually 3 different alleles that segregate in the human population. So one is called the IA allele, one is called the IB allele, and one is called the little I. IA and IB produce their respective antigens. So if you have the IA allele, you produce the A antigen. If you have this allele, you produce the B antigen. And if you have the little I, you don't produce either. But what this creates is an interesting set of relationships. So again, I previously discussed, and this is kind of a real life example, you always have to consider each combination of alleles to determine how they interact with each other. So IA and IB are dominant to little I. So this is dominant to this allele, and then this is dominant to this allele. But these two alleles have a codominant relationship to each other. So these are codominant. And so what does that mean? So if we look at the possible genotypes, all the possible genotypes, because A is dominant to little I, the phenotype has to be the same. And so they both produce the antigen A, and your blood type is A. Same thing with B, they have the same. But now in this case, IA and IB, you actually produce both antigens. And so that is the combination that leads to the AB genotype, the AB blood type, the AB phenotype. And only if you have both little i's do you not produce an antigen, and you would have the O blood type. So this is the genetics of this, and we're going to explore in the activity a lot more how you use this genetics and how you can use this to do things such as paternity testing. All right, the final example of, I'm not even sure how exactly to call this, but it's an interaction that occurs between two alleles that actually results in lethality. So this was identified in the studies of a mouse mutant that led to a yellow coat. So the wild type mouse had this agouti colored coat, and they isolated mutants that had a yellow coat. So the color changed. But the genetics were very, very weird. And so what they found when they crossed an agouti with a yellow coat individual was this weird ratio of 1/3 agouti and 2/3 yellow. And so this did not make any sense based upon the known rules of inheritance. Usually we always see it as a function of like 1/4. And so what it was realized is that there are two alleles. So there's the agouti and then there's the yellow allele. But what happens is that the homozygous for the yellow allele results in dead mice. And this death occurs during development of the fetus, and so you don't actually observe any of these mice. So these mice die very early on in development, and so when a mother has the pups, it's not obvious that a proportion of the animals have died. And so if you do this, normally this would be 1/4. Normally this would represent 1/4. Normally this would represent 1/4, and then normally this would represent 1/4. And so if you added these two up, it would be 1/2 yellow, 1/4 agouti, and 1/4 this genotype. But now because they die, you do this procedure called renormalization. So we've seen this before in the chapter 3, where we looked at pedigrees, and in the activity we talked about renormalizing probabilities based upon the ability to exclude genotypes, and the concept here is the same. And so you can exclude these genotypes, because you can exclude this as a possibility, you simply have to renormalize these probabilities. So you add up all of the remaining genotypes, and those add up to 3/4. And so you divide these numbers by this in order to figure out the final ratio. So agouti would be 1/4 divided by 3/4, which is where you get the 1/3, and then the yellow would be 1/2 divided by 3/4, which is where you get the 2/3. So whenever you see a ratio of thirds, you should immediately think to yourself, oh, this might be a lethal gene.